Hello, everyone, and welcome to this second screencast lecture on muscles and muscle tissue. In this in this lecture, we're going to take a look at the physiology of muscle contraction. So we're going to answer the third question of anatomy and physiology. Remember, when you study anatomy and physiology, there's always three questions that you want to answer. The first question is, what's there? What are the organs and structures that make up the human body, maybe system by system? Number two, what does what's there do? And that's basically functional anatomy. And then the third question that we ask is, how does it do what it does? And that's what we're going to take a look at for muscles and muscle tissue now. We're going to take a look at how muscle contraction takes place. All right, to understand how muscle contraction takes place, then we're going to have to take a look at some background information first and look at three important concepts. The first is the concept of transmembrane potential. You may remember that from A&P 1. The second concept is the concept of what we call the motor unit, and our third concept is what we call the neuromuscular junction, that place where a neuron that innervates, a motor neuron that innervates or supplies a muscle, meets the actual muscle fiber itself. That's referred to as the neuromuscular junction. We'll get into more detail on that as we move along. But first of all, though, let's take a look at the concept of transmembrane potential. And again, you've already studied this in A&P 1, so we're just going to kind of review things. Remember that transmembrane potential is the charge difference or voltage that's maintained between the inside and outside of a cell membrane. So if we look at our little diagram of a cell here, we can see that with regard to transmembrane potential in irritable cells, that is cells that have the ability to respond to a stimulus, that we have an overall net positive charge along the outside of the cell membrane, the plasma membrane, and along the inside of the plasma membrane, there's an overall net negative charge. Now, this difference in charges is what we refer to as the transmembrane potential, and what's responsible for that primarily on the outside is the fact that the cells don't like sodium ions. So they tend to, when sodium ions are in a higher concentration, try to enter the cell tends to reject those sodium ions. Sodium ions are positively charged. They accumulate right along the outside of the plasma membrane of the cell, and the cell gets an overall net positive charge. On the inside of the membrane, this negative charge is due to the presence of proteins, which have a net negative charge along their backbone at somewhere around neutral pH, which the pH of the inside cell is close to neutral, and and also the presence of negatively charged ions. So this is transmembrane potential. The second concept is the concept of the motor unit. A motor unit is defined as the motor neuron plus all of the muscle fibers that motor neuron stimulates. So we can take a look down here. Here is a motor neuron that's exiting the anterior horn of the gray matter of this section of the spinal cord. And if we just follow this one axon, here's the cell body. And then we have the axon, the appendage, the long extension that extends out. And we can see that it branches. And then this axon at its terminus, this axon is associated with this muscle fiber. And the terminus of this axon is associated with this muscle fiber. Now, if we take a look at this next axon here, we follow it along. We can see it gives off branches. And those branches excuse me, innervate or supply this muscle fiber and this muscle fiber and this muscle fiber. So three muscle fibers innervated or supplied by the axon of this motor neuron. So that the axon of that motor neuron plus the three muscle fibers it innervates makes up a motor unit. On the other hand, we look at this first motor neuron that we looked at, we can see that it controls these two fibers. So these two fibers plus the axon of the motor neuron that controls it makes up another motor unit. So we can see that there are actually two motor units detailed here. By the way, if we look over here, this is an actual micrograph taken with a light microscope showing the axon, and we can see the branches, the telodendria of the axon, and we can see the axon terminus for each one of them, and we can see how this one's supplying this fiber, this one's supplying this fiber, and so on. So we're seeing actual motor units there as well. So that's the concept of the motor unit. And the third concept is the concept of the neuromuscular junction. So a neuromuscular junction is a small gap that exists between the end of a motor neuron and the sarcolemma. So recall, if you haven't had me for A&P1, one of the things I always kind of stress in A&P1 is that we're made up of some 60 to 100 trillion cells in our body, depending upon the size of our body. But no two of those cells really ever touch one another, well, with the exception of some epithelial cells, which have kind of tight junctions where they're kind of spot welded together, you may recall from A&P1. But even those cells have little gaps in other places between them. They're just kind of spot welded together at tight junctions. So the cells always have a little gap between themselves and the next cell, and sometimes the gap is even bigger. Well, the same is going to hold true whenever we talk about neurons that supply muscle fibers. The neuron comes close to, if this hand is the sarcolemma, or the plasma membrane of the muscle fiber, and this represents, my fist represents the end of the axon, or the terminus. The axon terminus is not going to touch physically the sarcolemma, but rather it's going to come very close to it, and there's always going to be a little space between there. That space, that small gap where the neuron meets the plasma membrane, the sarcolemma of the muscle fiber, is called the neuromuscular junction. So when a nerve impulse, a charge reversal, you may recall from A&P1, reaches the end of a motor neuron, what happens? You may recall that the motor neuron has little packets of chemicals that are in the terminus, and these chemicals are referred to as neurotransmitters. When the nerve impulse, the traveling charge reversal, the depolarization reaches the axon terminal, the axon terminal will then release this chemical neurotransmitter into the surrounding environment. In the case of a neuromuscular junction, that neurotransmitter is almost always acetylcholine, abbreviated ACH. So when a nerve impulse reaches the axon terminal that lies next to the sarcolemma, the terminal is going to release acetylcholine, the neurotransmitter. Once the acetylcholine is released into what is referred to as the cleft, the acetylcholine then will diffuse across the cleft, or across the neuromuscular junction, from one side of the junction to the other, and the chemical acetylcholine will now attach to receptors on the surface of the sarcolemma, that is to say, the plasma membrane of the muscle fiber. As a result, the sarcolemma will become temporarily permeable to sodium ions. The sodium ions will then rush to the inside of the muscle fiber. Now remember, the sodium ions are accumulated in abnormally high concentrations just outside the sarcolemma, because sodium ions that tried to get in were kicked back out. They're in a higher concentration outside the sarcolemma than they are inside the sarcolemma. And if you remember the law of diffusion, they want to move from where they're in high concentration to where they're in low concentration. But something in the membrane is blocking them from getting in. And we know what that something is. There are chemically gated channels, sodium channels, in the sarcolemma that would allow sodium to pass, but the gates of those channels are closed. That is to say, until the neurotransmitter acetylcholine is present. So when acetylcholine is present, if my hand is the receptor on the channel, then the acetylcholine will bind to that receptor. Once it binds to the receptor, that will open the gates of the sodium channels and allow the sodium ions to rush to the inside, as we're saying here in this slide. When the sodium ions rush to the inside of the cell, what's going to happen? There's going to be a reversal of charges. So the loss of the sodium ions from the outside and the gain of sodium ions to the inside causes now the inside of the cell to become positively charged, while the outside of the cell now becomes negatively charged. And it's negatively charged because we lose positive charges, but no negative charges are lost from the outside. When we reverse the charges this way, this is referred to as an action potential. So hopefully you learned about more detail of the formation of action potentials in A&P 1. So here's a diagram that represents what's going on here, what we kind of just explained. So this is the terminus of a motor neuron, the axon of a motor neuron. And here we can see these little packets of chemical. These packets of chemical are synaptic vesicles. They contain the neurotransmitter acetylcholine. Now notice that the motor neuron, the terminus of the motor neuron, lies near the sarcolemma of the muscle fiber, but doesn't actually touch it. So here is the sarcolemma, the plasma membrane of the muscle fiber. Nowhere in there is that axon terminus touching the sarcolemma. So you can imagine if a charge reversal, a depolarization, an action potential reaches this terminus, what's going to happen? That's going to stimulate synaptic vesicles in the terminus to begin to move toward the plasma membrane of the axon terminus, fuse with that membrane, and release the neurotransmitter acetylcholine into the space between the membrane of the neuron and the sarcolemma. This space is referred to as the synaptic cleft. What will happen? Well, the acetylcholine being in a higher concentration on the neuron side of the cleft and a lower concentration on the muscle fiber side of the cleft will diffuse from where it's in high amount to where it's in low amount across the cleft, and eventually, as we can see here, the neurotransmitter will bind to receptors on the surface of the cleft. Okay, so that's where we're at to this point in time. Okay, so now what happens once that takes place? Let's take a look now at what's going to happen. What are the steps of a muscle contraction? Here's what we already know, what we just talked about. When an action potential reaches the axon terminus, the axon terminus is going to release acetylcholine from synaptic vesicles into the neuromuscular junction or the cleft. The acetylcholine will diffuse across the synaptic cleft and will attach then to receptors on the sarcolemma. As a result, the sodium channel gates will fly open and sodium will rush to the inside of the muscle fiber. This will result in the transmembrane potential of the sarcolemma being reversed, and it creates an action potential. So an action potential now is a charge reversal in which instead of there being positive outside, negative inside, the cell now along the membrane is negative outside and now positive inside. All right, so what's going to happen next? Well, once the action potential forms on the surface of the sarcolemma, just like along axons, an action potential acts as a stimulus to create new action potentials all across the surface of the membrane. And so what happens? The action potential begins to spread across the surface of the sarcolemma. All across its surface, the sarcolemma begins to depolarize. The areas of depolarization are called action potentials. So now, eventually, the action potential is going to enter the fiber through T tubules. So you recall in our first screencast, we mentioned that there were these perpendicularly running tubules that opened to the sarcolemma. These are the T tubules. The action potential as it spreads across the surface of the sarcolemma is eventually going to come upon a T tubule. T tubules are irritable as well, and they will respond to the stimulus by continuing to propagate the action potential, allowing the action potential to spread down through those T tubules. Eventually, the action potential will, as it spreads down through the T tubules, it will now then spread to the sarcoplasmic reticulum. Now remember that the sarcoplasmic reticulum has those cisternae that have large amounts of calcium in them. In the cisterns, or cisternae, there are calcium pumps that constantly pump calcium that might enter a muscle fiber out of the cytoplasm and maintain it inside the cisterns. But when an action potential spreads over the sarcoplasmic reticulum, calcium ions are released into the sarcoplasm, that is to say, the cytoplasm of the muscle fiber, by the sarcoplasmic reticulum. Why does that happen? Well, as I said, inside the sarcoplasmic reticulum, there are calcium pumps, and they're constantly turned on, and you could hear them humming along as they are pumping calcium, any calcium that enters the muscle fiber, pumping it immediately out of the cytoplasm and holding it in these cisterns. We don't want any free calcium just kind of running around inside the cytoplasm. We'll see why as we continue our discussion. But these pumps are on. But when an action potential spreads across the sarcoplasmic reticulum, it's sort of, you could think of it as short circuits those calcium pumps and turns them off. When the calcium pumps turn off, we think about it now, there is going to be a high concentration of calcium inside those cisterns and a low concentration of calcium in the sarcoplasm, in the cytoplasm of the muscle fiber. So what's going to happen? The law of diffusion is going to kick in, and the calcium ions are immediately going to diffuse out of the cisterns and into the sarcoplasm. And initially, they're going to do that very rapidly as they move from area of high concentration to areas of low concentration. Once the calcium ions leave the sarcoplasmic reticulum, they will immediately be picked up and they will bind to a special protein called troponin. And if you're having trouble following this, that's okay. Stick with me here. We'll show you a diagram of all this as we go along. Troponin is a protein that's associated with actin filaments that has a high affinity for calcium. So when the calcium is released into the sarcoplasm, the troponin immediately picks up the calcium and binds it. That binding of calcium to the troponin engages now what we refer to as the sliding filament hypothesis. And in the sliding filament hypothesis, remember there are two myofilaments that are associated with the muscle fibers. actin and myosin. Let's take a look at myosin first. Myosin, remember, is the thicker of the two myofilaments. It has cross bridges on it that could stand up off of it, kind of like my arms with a fist in the end. They have a head at the end. And these cross bridges have the ability to link on to actin. Okay? And what will happen is, in the presence of the calcium ions bound to the troponin, myosin cross bridges will attach to the actin. Once the cross bridges attach to the actin filaments, the cross bridges then will begin to ratchet and they will pull actin in over the surface of the myosin. As a result, remember, actin is attached to the Z lines of the sarcomeres. So as a result, if you pull the actin in, you're going to pull the Z lines in and the sarcomeres are going to begin to shorten. If the sarcomeres within a muscle fiber shorten, that muscle fiber is going to shorten. If muscle fibers inside a muscle shorten, that muscle is going to shorten as well. Okay, so let's say that we want a muscle to undergo contraction. We stimulate it with action potential from a motor neuron. The action potential spreads across the surface of the muscle fiber, enters the muscle fiber through the T tubules, spreads across the sarcoplasmic reticulum, and short circuits the calcium pumps. Calcium floods into the sarcoplasm, the cytoplasm of the muscle fiber. Calcium is released. The calcium binds to troponin, and that engages the sliding filament hypothesis. Myosin cross bridges pop up, they ratchet, and they pull the actin in over the myosin, which effectively shortens the sarcomere. As a result, what's going to happen? The muscle is going to undergo contraction. Let's say, for example, that my head is itchy and I want to scratch it. So I'm going to develop an action potential on the motor cortex of my brain. It's going to travel down over motor neurons. It's going to exit the spinal cord at a site associated with that arm muscle. That motor neuron is going to enter the muscle, give off the branches, and the action potential is going to be delivered to the muscle fibers. What's going to happen? The muscle fibers are going to contract. I'll be able to lift my arm, my forearm up to my head, and I'll be able to scratch my head. Now the itch is over and I want to put my arm back down. In order to put my arm back down, I have to have this muscle, which is now contracted, I have to have it relax and return to resting length. So that's what we're going to take a look at now. To get the muscle to return to resting length, the nerve stimulus has to be removed. When we remove the nerve stimulation, what's going to happen? The sodium pumps, you may recall the sodium potassium exchange pumps, are going to kick back on. They're going to pull the sodium back out, and the nerve impulse, or I should say the action potential, is going to flip back to a polarized state. As a result, when the action potential is removed, there's nothing short-circuiting the calcium pumps now in the sarcoplasmic reticulum cisterns. The pumps kick back on and they pull the calcium back out of the sarcoplasm. The calcium re-enters the sarcoplasmic reticulum. It detaches from the troponin and re-enters the sarcoplasmic reticulum. As a result, the myosin cross bridges will release the actin, so they'll pump down off of the actin, and the actin will now slide back out to its original resting position. The sarcomere will lengthen, the muscle fibers will lengthen, the muscle will relax. Okay, to help you to better understand this, let's take a look at how the process actually occurs. I'm going to show you an animation. This animation is also in Canvas, and you'll be able to take a look at it there. Okay, so let me just bring this up. I guess I'll have to allow Adobe Acrobat. There it comes. So this is a diagram that's going to represent an animation that's going to represent how the sliding filament hypothesis occurs and give us more detail on things. So let's just kind of orient ourselves. This is a muscle fiber, a muscle cell within a muscle proper, right? Inside the muscle fiber, there are these canisters called myofibrils, these proteins called myofibrils. And within the myofibrils, there are myofilaments. We can see there are alternating thick myofilaments called myosin and thin myofilaments called actin. And these myofilaments make up the sarcomere that runs from Z line to Z line. So this is showing Z line to Z line here. Now, lying over top of the myofibrils then, we're going to have, as we've seen before, here are T tubules. Here's the sarcoplasmic reticulum, and we can see the swellings in the sarcoplasmic reticulum where the tubules kind of fuse together to form these cisterns. It's in these cisterns that is where the calcium ions are being held. Okay? All right, so next what we're going to do is we're going to add an action potential to stimulate the surface of the muscle fiber. Okay, so let's let this run through and then we'll talk about what's going on here. So here we have up here, this is the synaptic terminal of a motor neuron. An action potential just traveled down this motor neuron. The synaptic vesicles moved to and fused with the plasma membrane of the motor neuron, what we sometimes refer to as the presynaptic membrane. They bust open and release the neurotransmitter acetylcholine. The acetylcholine binds to the surface of the sarcolemma, opens up the sodium channels, and sodium rushes to the inside of the cell across the plasma membrane, causing the muscle cell, the sarcoplasma or the sarcolemma, to become depolarized. Now we saw arrows here. Those arrows represented the action potential of the area of depolarization moving across the membrane until it hit a T tubule. Then what happened? The action potential traveled or was propagated into the T tubule, or what we also know as the transverse tubule. And then it was propagated across the sarcoplasmic reticulum. So here's the blow up of the sarcoplasmic reticulum. That causes the depolarization of the sarcoplasmic reticulum, sort of short circuits the calcium pumps, and causes calcium release from the sarcoplasmic reticulum. So these diamonds are going to represent the calcium. Okay. So next, let's move to the inside of the myofibril and let's take a look at the myosin and actin filaments. So here in blue, this is a myosin filament. Myosin, remember, is the thick filament. And here are those cross bridges or heads that we were talking about that are associated with the myosin filament. Here, this is the thin filament, actin. Actin is comprised of these gold spheres right here. And we'll notice that these gold spheres have little indentations on them. These indentations are active sites where myosin heads could actually link onto the actin and bind with the actin. But what do we notice? We notice there's this thin ribbon of protein that kind of wraps around the actin molecules. This ribbon of protein is called tropomyosin. And notice that the ribbon is covering all of the active sites on the actin. This prevents the myosin cross bridges from being able to attach to the actin. Notice also that there are these patches, lighter green patches of a protein called troponin that is attached along the ribbon of tropomyosin. Now, in the relaxed state, this is what we see, the organization. By the way, this is kind of the area where the actin partially overlaps the myosin. Okay, so this is in the A band, and an I band would be out here where there's actin only, and this is where out here where the the Z membranes would be. Okay, so the actin is attached to Z membranes out here. Now, when a contraction takes place, what's going to happen is the actin is going to slide, let's say in this direction, toward the center of the sarcomere, over the surface of the myosin. Now, how do we get the actin to slide over the surface of the myosin? Well, these cross bridges have to link on and ratchet, and as they ratchet, they'll pull the actin over the surface of the myosin toward the M line. Okay, and that will effectively shorten the sarcomere. But that can't happen right now in this relaxed state because the active sites are covered. So how do we get the active sites to uncover? Well, remember, the action potential reaches the sarcoplasmic reticulum. The action potential now sort of knocks out the calcium pumps, short circuits them, and calcium is going to be released into the sarcoplasm. This troponin has a high affinity for calcium, so the calcium now binds to the troponin. So let's add calcium in and see what happens. Here comes the calcium, it binds to the troponin. Notice what happens, it changes the shape of the troponin. The troponin pulls on the tropomyosin, and that tug on the tropomyosin uncovers these active sites. As a result, we notice these cross bridges now can bind to active sites, and they begin to ratchet and pull the actin in over the myosin toward the M line. Now, we should note here that once this cross bridge attaches to the binding site, it's sort of spring loaded and it automatically uncocks. So it attaches, it automatically uncocks and snaps to pull the actin in, as you're seeing there. Now, notice that the cross bridges constantly get recocked so they can perform that action of pulling the actin in over the myosin. In order to do that, that's going to require energy to recock that head. And that's what we're looking at here inside the circle. Here's a cross bridge, and coming in yellow, this is going to represent ATP. So what's going to happen is the ATP is going to be hydrolyzed. It's going to break down into adenosine plus a phosphate group now. This is, as a result of this, a bond is going to be broken, and when the bonds are broken, energy is going to be released. The energy that's released is used to recock that head so that it can continue to slide the actin over the myosin. Now, this is basically showing one side, but if we're thinking about both sides of the sarcomere, my hands representing the Z lines, the actin's going to slide in over the myosin from one side and the other side at the same time. So the distance between Z lines is going to shorten. The sarcomere is going to shorten. When the sarcomeres shorten, the myofibrils shorten. When the myofibrils shorten, the fiber shortens. When the fibers in a muscle shorten, the muscle shortens, and what happens? I'm able to move my hand up toward my head. Got it? Okay. So now let's take a look at relaxation. So I've moved my hand up. Now I want to let my arm fall back away from my head. To do that, I have to remove the calcium ions. So to remove the calcium ions, I'm going to remove the stimulus. that started it all off, the action potentials. The calcium pumps in the sarcoplasmic reticulum are going to turn back on, and that's going to pull the calcium back out of the sarcoplasm. So let's pull the calcium back out off the troponin. When that happens, the troponin returns to its original shape, stops tugging on the tropomyosin. The tropomyosin now covers the active sites again. The cross bridges cannot interact with the active sites. So as we've seen, the actin slides back out over the myosin and the sarcomere lengthens. Where am I at here? There we go. When the sarcomere lengthens, the myofibrils lengthen. When the myofibrils lengthen, the fiber lengthens, relaxes, and the muscle relaxes, and I drop my head back down. So this is a very good animation to help us to understand what's going on there. And I hope that helps you to understand. And again, this animation is in Canvas under videos and animations so that you can study it when you're preparing for the test. Okay, so back to our PowerPoint, let's continue on from there. Okay, so let's take a look at rigor mortis. And rigor mortis literally comes from the Latin rigor meaning stiff and mortis meaning death. Okay, so rigor mortis actually refers to the state of the body after death in which the muscles become stiff. This usually begins approximately 3 hours after death, and the muscles will reach their maximum stiffness in approximately 8 hours. But then the stiffness will gradually dissipate until about anywhere from 24 to 36 hours post-death. Now, why does this occur this way? Well, from what we just talked about, you should be able to understand this. So what's going to happen when an individual passes? The cells can still continue to manufacture ATP for about 3 hours via anaerobic respiration. Not aerobic respiration, but only anaerobic respiration now. As a result, ATP can be produced for, as I said, 3 hours, and the ATP that's produced provides the energy to maintain the calcium pumps in the sarcoplasmic reticulum. But after 3 hours, the ATP begins to dissipate because it can't be produced anymore. So as it's used up, you can't keep making more ATP. Once all the ATP is used up, there's nothing to maintain the calcium pumps, and calcium begins to leak into the sarcoplasm from the sarcoplasmic reticulum cisternae. And what happens? When the calcium begins to leak out, it attaches to troponin, pulls on the tropomyosin, opens up the active sites, the myosin cross bridges pop up and begin to ratchet the actin in over the myosin, pulling the actin in, and as it does, the muscles gradually stiffen. Okay, now remember, in order to be able to ratchet in, you might be thinking, don't they need ATP? Yes, they do. The myosin cross bridges need ATP, but myosin has its own supply of ATP stored right on the myosin filament itself. So that's where the ATP is going to come from for those myosin cross bridges to ratchet. Eventually, what's going to happen is the ATP is going to be used up and there's nothing to recock the heads. But the calcium is still present, so the heads are still going to be bound to the active sites, but they can't be recocked. So do you follow along? The actin slides in, and eventually there's no mechanism to release the actin or release the myosin cross bridge from the actin, and so the actin stays permanently pulled in over the myosin and the muscle stays stiff. This is going to, again, maximum stiffness is going to be reached at about 8 hours, but gradually it's going to dissipate until about 24 to 36 hours post-death. So at 24 to 36 hours post-death, everything's going to be relaxing. And what's going on there? Well, that relaxation is not due to uncocking the heads, but rather it's due to the fact that lysosomes inside the muscle fibers now burst open. They release their digestive enzymes, and the actin and myosin begin to be digested. As the actin and myosin are digested, then the muscle begins to relax. So this is in forensic science, this is how the time of death can help to be determined. So whenever you think about it, if the muscles aren't stiffened yet, then the person hasn't been dead for approximately 3 hours. If they've been dead from 3 to 8 hours, the muscles will be in a certain state of stiffness. And if the muscles are relaxed again, then the individual would have been dead for more than 24 hours, up to 36 hours. Okay. Now you might be looking at that and going, well, how do you know the difference between when the muscles are not stiff and you're in those first 3 hours versus when you're in those 24 to 36 hours post-death? Well, that's where temperature comes into play. Because as ATP is being broken down, the energy is released to recock the heads, but not all the energy is necessary to recock the heads. As a matter of fact, 60% of the energy is released from the breakdown of ATP is given off as heat. And that's what helps to keep our body temperatures warm. So you can imagine that when the ATP supplies are all done, ATP is not being broken down anymore. There's nothing to continue to generate body heat. And what will happen is the body temperature will gradually go down until the body temperature is the same as the surrounding temperature. So if the body temperature is the same as the surrounding temperature and the muscles are relaxed, we're at that 24 to 36 hour window. If the body is still warm, comparing it to the surrounding temperature, then we'd be in that first 3 hours post-death. So I thought that would be interesting to understand how we can put what we're learning in anatomy and physiology together to understand a situation like rigor mortis. And again, there it is. The dissipation occurs as the lysosomes begin to digest the muscle proteins and especially the actin and myosin. Okay, next up, I'd like to talk a little bit about how muscles grow and develop. First of all, with regard to muscle growth, there are actually two ways that muscles grow. One way is that the muscles stretch. Remember, muscles exhibit elasticity. Uh, they can be stretched. So muscles have two sites of attachment that we'll talk about in our PowerPoint on this, our second PowerPoint on the muscular system, or second chapter, I should say, chapter 10 on the muscular system. And we'll see that they have two attachment sites, one proximal, one distal, and oftentimes those muscles will cross a joint. So when the bones actually grow, if we have an attachment site here on one bone and an attachment site here on another bone, as the bones grow in length, that's going to stretch the attachment sites out. And the muscle, because it's elastic, will stretch as the bones grow in length. But the other way that muscles grow is by increasing in diameter due to the addition of actin and myosin filaments that are already present in existing muscle fibers. So we're going to add more actin and myosin filaments to already existing fibers. So one of the keys here is that muscles don't primarily grow by adding more fibers. You know, you have approximately today, as you're sitting there, the same number of muscle fibers in your body that you had the day that you were born. The difference is those muscle fibers have been stretched and their diameter has increased as more actin and myosin filaments are being added into those already existing fibers. So understanding how they're stretched is pretty simple, but how do we add actin and myosin to already existing muscle fibers in order to get the muscle to grow in diameter? Well, let me give you a little bit of background first to help you understand this. And you should have this background already in your PowerPoint without any blanks in it. If you have a few blanks, you can fill them in, but by and large, you should just be able to follow this along. So the energy for muscle contraction is provided directly by ATP. We saw that in the previous animation on the sliding filament hypothesis. But as ATP is depleted during contractions, it has to be resupplied, restocked, reorganized, so to say. Now, how is ATP reorganized? Well, it can be reorganized through the process of cellular respiration, but cellular respiration, relatively speaking, is kind of a long, kind of arduous process. Takes too long. And if we're currently in a state where we're asking muscles to maintain a state of contraction, if we run out of ATP, reorganizing more ATP via cellular respiration can take too long. So the muscle has another method built in to reorganize ATP very quickly, and that is it uses creatine phosphate, or CP. Creatine phosphate molecules can transfer phosphate groups to ATP, thus regenerating, or excuse me, to ADP, thus regenerating more ATP right there on the spot, right at the site where the muscle contraction is taking place. However, that chemical reaction that transfers a phosphate group from creatine to ADP is going to result in the accumulation of free creatine inside the muscle fiber. The fiber then is going to recognize this accumulation of free creatine as a threat to its ability to meet its energy demands. So what we're saying here is the more free creatine that accumulates inside the muscle fiber as a result of contractions, the muscle goes, wow, look at all that free creatine. We almost ran out of the ability to reorganize ATP. So to compensate, the muscle fiber will organize and store additional creatine phosphate supplies so that the next time we exercise, we're going to have larger supplies of creatine phosphate to reorganize ATP in the hope that we wouldn't run out as quickly. However, when you manufacture creatine phosphate and you want to keep it there as an on-demand supply, you have to have a place to store it. The place to store creatine phosphate is right on the myosin filaments along with ATP. Thus, if you're going to manufacture more creatine phosphate, greater supplies of creatine phosphate for the next bout of physical exercise, you're going to have more storage containers, i.e. myosin, to put the creatine phosphate in once you manufacture it. So more myosin is going to have to be created. And in muscle fibers, along with myosin, is always created actin. So exercise, which depletes creatine phosphate and creates free creatine supplies, causes the muscle to create more free creatine and at the same time increase the number of myosin and actin filaments inside the muscle fiber, and the muscle fiber is going to get thicker. Again, let's take a look at this in terms of a diagram. So here we have an individual who is going out for a run and using these large leg muscles whenever he's going out for a run. So contraction of the muscles that allows him to run depletes ATP supplies. When the ATP supplies become depleted, creatine phosphate then is going to be used to reorganize the ATP to continue the muscle contractions. But as this phosphate group is transferred from creatine phosphate to ATP, that creates free creatine inside the muscle cytoplasm. This free creatine then will act as a feedback mechanism to stimulate the muscle to produce RNA, messenger RNA. So the free creatine stimulates the production of messenger RNA. Now you may recall that messenger RNA acts as a blueprint for building proteins. What proteins are we going to want to build? We're going to want to build more myosin and actin. Why? Because we're going to want to, if we got this free creatine here, we're going to want to respond to that free creatine not only by producing the messenger RNA, but by producing more creatine phosphate supplies. We're going to need myosin and therefore actin to store the additional creatine phosphate supplies. So ribosomes will manufacture myosin and actin inside the muscle fibers. The muscle fiber, or the myosin and actin, will be added to the muscle fiber. By the way, as actin and myosin are added to the muscle fiber, you now have more pooling elements in the sarcomeres. So when the actin is pulled over the myosin, you now can create stronger muscle contractions because you have more pooling elements. Okay? But with those stronger muscle contractions, there's going to be the breakdown of even greater supplies of ATP, which is going to require even more creatine phosphate to be broken down, which creates even more free creatine, and this results in a cycle of events that keeps occurring over and over. So as it occurs, more and more actin and myosin are being added to the muscle belly, to the muscle fiber, I should say. Okay, so this is why, for example, in youngsters, play is essential. Because what does play do? Play causes muscle contractions, burns up ATP, results in the production of free creatine, and causes muscles to grow as actin and myosin are added to them. Now, there's a couple of other points that we'd like to make here, and this is, by the way, this cycle that I just showed you is called the creatine phosphate cycle of muscle growth. A couple of other points that we'd like to make here is that over the course of time, researchers have studied this and said, hey, that's interesting. So if free creatine accumulates in a muscle and that stimulates the production of messenger RNA, which results in myosin and actin being added to the muscle and the muscle growing bigger and stronger, what if we had an individual take free creatine, drink, let's say, for example, creatine, and have the creatine absorbed into the muscle? So now, instead of just a little bit of free creatine being created when the muscle undergoes contraction, you get a lot of free creatine accumulated in the muscle. Will this stimulate an increase in even more messenger RNA production and therefore more myosin and actin? Well, as it turns out, adding more free creatine during workouts can modestly increase the production of messenger RNA and therefore increase myosin and actin production, which can modestly get the muscle to grow bigger and stronger a little bit faster. But there are some downsides with that. Taking the free creatine causes a kind of reverse osmosis that can draw water out of muscles. And as you draw the water out of the muscles, remember, most of the chemical reactions that occur inside our cells require water. So that's going to be counter to the process here. And that's why I say it's a very modest increase in myosin and actin. On the other hand, you hear about steroids, anabolic androgenic steroids like testosterone and testosterone type of drugs that can be used. Do they work? Actually, steroids work, and here's where they work. Normally, in this creatine phosphate cycle, if you get a little bit of free creatine being produced from muscle contraction, you'll stimulate a little bit of messenger RNA production, which will result in a little bit of myosin and actin added to the muscle, causing the muscle to become bigger and stronger. But under the influence of steroids, what happens is a little bit of free creatine doesn't result in a little bit of messenger RNA production. It instead results in a burst in messenger RNA production. So you get a burst of myosin and actin added to the muscle, so the muscle gets bigger and stronger faster. Now, this does require exercise. It does require the muscles to be used to create the free creatine in the first place. If you just give an individual androgenic anabolic steroids and you don't have them exercise their muscles, they're not going to result in the addition of myosin and actin. But with exercise and steroids, there's going to be a lot more myosin and actin added to the muscle to get it to grow bigger and stronger faster than it would otherwise under normal circumstances. Okay? Okay, along with muscle growth and development, then a couple of terms I'd like to talk about. The first term is hypertrophy. So when a muscle increases in size, and this happens as a result of activity, so undergoing muscle contraction or using the muscle, this increase in size is referred to as hypertrophy. The muscle, when it increases in size, is said to hypertrophy. On the other hand, if we don't continue the contractions or continue to use the muscles will respond by saying, well, he's not asking me to do this anymore. Why do I have all this extra actin and myosin sitting around here? And so what will happen is the additional actin and myosin, usually about, if you don't continue to use the muscles at about 92 hours, actin and myosin will gradually be removed from the muscle, again by the lysosomes. And this will cause a loss in the size and the strength of the muscle due to that disuse, and that's referred to as atrophy. Okay, next up, let's take a look at some types of muscle contractions. So at the beginning of this, we had a little asterisk under contraction. We said a muscle contraction was the result of the shortening of the muscle developing force. Well, now we're going to adjust our definition a little bit here and say that muscle contraction doesn't always necessarily mean that the muscle contracts or shortens. And we're going to adjust our definition of a contraction to say that a muscle contraction now is the generation of tension or pulling force. That's because the muscle's not always necessarily going to shorten when contraction takes place, but tension or pulling force will be created. This tension develops when actin and myosin filaments interact and at least attempt to slide past one another within the sarcomeres. With that in mind, we can look at what we call isotonic muscle contractions. These are changes in muscle length, or these types of contractions occur, I should say, when the muscle changes in length and moves the resistance. So the muscle actually does shorten and it pulls and moves the resistance. Let's say, for example, if I have my hand out and I want to move my hand up to my head, the weight of my hand could be the resistance. And if I want to contract and pull my hand up to my head, the muscle is going to shorten. It's going to pull against the resistance, and it's going to move the resistance, my hand up to my head. When that happens and we get movement that way, those are called isotonic contractions. So isotonic contractions are going to result in movement. Now, isotonic contractions can be broken down into two phases, a concentric phase and an eccentric phase. Concentric phase or concentric contractions occur when the muscle shortens as it develops tension to overcome the resistance. So as I'm moving my hand up toward my head, the muscle is actually shortening. My bicep muscle is actually shortening, and my hand is moving up towards my head. That would be a concentric contraction. On the other hand, eccentric contractions occur when the resistance overcomes the active muscle and the muscle lengthens as it develops tension. So for example, if I have my hand up like this in a slightly contracted state, and maybe I should turn sideways to show you this, so I'm like this, and if I put force against my arm and I add weight or put enough force against my hand that the muscle still maintains the attention or the tension, but gradually lengthens, then that's referred to as the muscle being in a state of contraction, but it's lengthening, so that would be an eccentric contraction. Okay, in addition, another type of muscle contraction occurs when the muscle develops tension but does not shorten. When a muscle develops tension but doesn't shorten, these are referred to as isometric contractions. So let's take a look at these diagrams and see if you can tell me what type, based upon that explanation. Let's see if I've done a good job. If you can tell me what type of contraction must be taking place here. So here in diagram A, we have an individual who's doing a dumbbell curl and raising the dumbbell from his waist up to his neck. What type of contraction would this be? Would this be isotonic or isometric? If you said isotonic, that's correct. Now, is this an eccentric or concentric contraction? Remember, as he's lifting it up, we're getting movement and the muscle is shortening. So with the muscle shortening and overcoming the resistance, this would be, right, concentric contractions. All right, here in this diagram, we have an individual standing in a doorway and pushing out against the door frame with his hands. What type of contraction would this be? Would the muscles be in a state of isotonic or isometric contraction? If you said isometric contraction, you would be right, because notice there's no way we're going to be able to push this door frame apart. So particularly these tricep muscles, for example, are going to contract and they're going to generate force, pulling force, but there's no way we're going to be able to push that frame apart. So those muscles are going to stay, are not going to shorten, but great tension is going to be created. That would be an isometric contraction. And here in our third diagram, again, we return to this time we have a dumbbell curl that we're doing, but this time the dumbbell is overcoming the contraction and the arm is gradually lengthening. What type of contraction would this be? If you said this would be also an isotonic contraction, that would be correct. And since the muscle is maintaining tension and letting the dumbbell down slowly, this would be eccentric. All right, next let's talk a little bit about muscle tone. So muscle tone is the slightly contracted state of muscles. Okay, so what happens is as we exercise muscles, nerves constantly will send signals to the muscles to keep them in a constant state of readiness. The more muscle fibers are stimulated within any particular muscle to maintain that state of readiness, the more taut or tense the muscles will be. This state of slightly contracted state or this state of tautness of the muscle is referred to as muscle tone. When a muscle is untoned, it is said to be flaccid. So soft, flabby muscles are what we call flaccid muscles. These result from decreased stimulation. So if you don't use your muscles to exercise or play over a certain period of time, they will begin to become soft and flabby. The muscles are said to be flaccid. And again, That's because of a decreased state of stimulation from the nervous system. Next up, let's talk about types of muscle fibers. So not all muscle fibers are the same. We basically have two general categories of muscle fibers: slow twitch muscle fibers and fast twitch muscle fibers. So slow twitch muscle fibers have a relatively slow speed of contraction. That's where they get their name from. Slow twitch muscle fibers are generally thin, but they do have a very rich blood supply. Because they have a very rich blood supply, they tend to be dark red in coloration, and so they're also known as red fibers. Now, slow twitch muscle fibers can contract for a prolonged period of time, but they generate very little power. Relatively speaking, whenever they contract, now the contraction is going to be rapid, but within a range, slow twitch muscle fibers tend to contract much more slowly. They have a rich blood supply, so they have great ATP supplies. So they don't generate a lot of force from contracting rapidly, but because they have large ATP supplies available to them, large oxygen supplies available for aerobic respiration, they can work for prolonged periods of time. Fast twitch muscle fibers, on the other hand, are, relatively speaking, faster contracting fibers, so they contract much more rapidly. Fast twitch muscle fibers, generally speaking, have a larger diameter than slow twitch muscle fibers, but they are generally paler in color. They don't have as rich a blood supply. Because they're paler in color, they are referred to as white fibers, although they're not white, they're kind of a pinkish coloration. But fast twitch muscle fibers, because of their fast contraction, can create very powerful contractions, but they tend to kind of poop out, so to say, very quickly because they don't have as rich a blood supply. They don't have as great an oxygen supply. They can't regenerate ATP as rapidly, so they don't work very well over the long haul. They poop out very quickly within a minute or so. So how does this play out in the real world? Well, here we have an individual on the left side of the screen as you're taking a look at it, and she's a marathon runner. Marathon runners will tend to have a higher percentage of slow twitch muscle fibers. World-class marathoners will have as much as 80% of their muscle fibers will be slow twitch muscle fibers. This is what allows them to go for long periods of time without their muscle fibers pooping out. But remember, they're not going to generate a whole lot of force. On the other hand, this individual on the right side of your screen is going to be a sprinter, let's say a 100-meter dash person. To be a world-class 100-meter dash individual, you're going to have a higher concentration of fast twitch muscle fibers. World-class sprinters will have as much as 60% of their muscle fibers will be of the fast twitch type. Those fast twitch muscle fibers are what give them the explosive force that they need to run down the track very rapidly, but they can't maintain that for very long. Those fibers are going to poop out relatively quickly. All right, that takes us to heat generation. So when muscles undergo contraction, the heat that's released during a muscle contraction is referred to as initial heat. Remember that in order for a muscle contraction to take place, ATP has to be broken down to recock those heads, right? Only about 40% of the energy that's released when the ATP is broken down is used to recock that head. 60% of the energy is lost to the surrounding environment as heat. So that heat that's produced while the muscles are undergoing contraction is referred to as initial heat. Here's a diagram, or here's a picture of Lamar Odom when he played for the Lakers, and you notice how he's sweating because he's producing a lot of heat from the activity of the muscles while he's playing his sport, and he's trying to dissipate that heat through perspiration. That heat that's being produced while he's active is called initial heat. On the other hand, we have what we call recovery heat. Recovery heat's the heat that's produced for up to 30 minutes after exercise. Recovery heat is produced as ATP supplies are restored after exercise is done. This restoration of ATP also results in a loss of heat to the surrounding environment. So, depending upon how strenuous the exercise was, the heat is going to be produced until those ATP supplies are restored, and that can be for up to 30 minutes. Now, if your exercise isn't that strenuous, then you may not burn as much ATP, so your recovery heat may not last as long. It might last only 15 minutes. But on the other hand, if you had very strenuous exercise, burned a lot of ATP, and really depleted your ATP supplies, then it might take up to 30 minutes to restore those supplies. And all that time, heat is going to be produced. So here we have a tennis player. He's now in recovery heat. His match is over. He's done, but he's still sweating as his ATP supplies are being restored because of the heat that's being left off, and he's trying to dissipate that heat. So initial heat and recovery heat. Okay, next up, we want to talk a little bit more about muscle mechanics and muscle stimulation and contraction. So with regard to muscle contraction, we can not only talk about isometric and isotonic contractions, whether or not the muscle is shortening as tension is being developed, but we can also take a look at types of contractions from the standpoint of nerve stimulation. So let's do that now. So one of the things we know, as we've been talking about, is that muscle tissue is irritable. It will respond to a stimulus. For muscle contraction, the stimulus is provided, as we've seen, by a motor neuron. The question is, how does the muscle respond to the application of the stimulus by the motor neuron? In a laboratory, stimulus can be provided by an electric shock so that we can study how the muscle is going to respond and bring it down to all the way to just one single electric shock at a time. So how does a muscle respond to electric shock and therefore to stimuli? Well, we can take a look at three types of contractions with regard to the application of the stimulus. The first type of contraction is called a single twitch contraction. A single twitch contraction is a simple contraction that results from the application of just a single electric shock. Again, we're doing this in a laboratory situation. This is the most basic form of muscle contraction. It's called a single twitch contraction. A twitch is a short, single, quick contraction and relaxation of the muscle fiber. Now, when this muscle fiber undergoes contraction as a result of a single electric shock, there are what we can refer to as phases associated with it. The first phase of the contraction is referred to as the latent period. The latent period is defined as the time between the application of a stimulus and the start of the contraction. For humans, the latent period is usually somewhere around 10 milliseconds, a very brief period of time. But what we're saying here is that once we apply the stimulus, the muscle just doesn't automatically respond by undergoing contraction. There's a little delay before the contraction takes place. That little delay is called the latent period. The next phase of single twitch contraction is called a contraction phase. That's the time during which the muscle shortens. For different muscles in the body, whether they're fast twitch or slow twitch, that contraction phase is going to take different amounts of time. But this is the actual phase when the muscle is actually shortening, those sarcomeres, and therefore the fibers are undergoing shortening. And then that contraction phase is immediately followed by a relaxation phase. The relaxation phase is the time from the removal of the stimulus to relaxation or return to original resting length. So we're going to see that we apply the stimulus. There's going to be a brief pause. Then there's going to be a contraction phase in which the muscle is going to shorten. And then that's going to be followed by a relaxation phase in which the muscle is going to return to resting length. Okay, we can plot this out on a diagram to maybe help you understand this a little bit better. On a diagram, we're going to put this blue dotted line on there, and then we'll have a red dotted line. Now, the blue dotted line is going to represent the latent period. So let's say, for example, we apply a single electric shock at time 0 right here. We know now from our studies that there's going to be this period of time in which the muscle does not respond. It does not immediately begin to contract. That takes about 10 milliseconds, and that's referred to as the latent period. Once that occurs, the muscle is now going to respond to the stimulus, and it's going to start to contract. It'll shorten, and as the muscle shortens, it'll generate force. So here on our y-axis, we're looking at the force of the contraction. So what's going to happen is the muscle is going to undergo a contraction, and as it does, it's going to create pulling force. This point in time in which the sarcomeres are shortening, the fibers are shortening, therefore the muscle is shortening, is referred to as the contraction phase. That contraction phase is then immediately going to be followed by a relaxation phase in which the muscle is going to relax. As a result, the sarcomeres are going to lengthen, the fibers are going to lengthen, the muscle is going to lengthen. And as that happens, gradually the force is going to decrease. So we're going to go back down on the graph. So this peak up here represents our magnitude or peak force or tension development. Okay? This entire, for a single stimulus, this entire single twitch contraction will take approximately what? As we can see in the graph, 100 milliseconds. So what if we apply not just a single electric shock, but multiple electric shocks in sequence to the muscle and the muscle fibers? Well, this is referred to as summation contraction, and it will result in a contraction of even greater magnitude or force resulting from the application of the second stimulus before the end of the contraction. Now, these multiple stimuli have to be applied at the correct time. So with regard to time, we look at what's called the absolute refractory period first. This is the time period during which if we apply a second electric shock to the muscle, it will not respond to that second electric shock because it's already in a state of contraction. The next time period we take a look at is called the relative refractory period. This is the time period during which a second stimulus, when applied to the muscle fiber, will bring about another contraction and therefore summation. So here's how it works. Let's look at our graph again. So here is our latent period. Here's our single twitch contraction. So we apply our first stimulus here at time 0. There's a 10 millisecond delay, and then the muscle undergoes the contraction phase. Now, the muscle then goes into a relaxation phase. Let's apply a second stimulus here during the relaxation phase in the relative refractory period. If we apply a second contraction there, the muscle will be stimulated to contract again. And when it contracts the second time, it'll contract, as we see here, with even greater force or tension being created. So it's such that applying the second stimulus adds a second contraction, and there's this additive effect, so we call it summation. Okay, should mention here that a single twitch contraction does not generate enough force physiologically to actually cause body movement. Neither does a summation contraction here when we apply just a second stimulus. Yes, the tension is greater, but let's kind of use this imaginary line right up here. It's still not great enough. This is where we'd have to get in order to be able to cause effective body movement. But we do get this summative effect. So the magnitude is increased. So what's the cause of the increase in that magnitude, in the increase in the force? Well, it's called the TREP or warm-up phenomenon. The TREP or warm-up phenomenon says a long series of summations will result in a stair-step increase in the magnitude or the pulling force that's generated. This is due to two things. First, it's due to heat production. As the muscle warms, as it undergoes these contractions, ATP is broken down, energy is released as heat, the muscle warms up, everything becomes more efficient. The enzymes that break down ATP and allow for these contractions to take place become more efficient. This usually occurs in the first 5 contractions. The muscle warms and becomes more efficient, so we get greater force. After those first 5 contractions, though, the increase in magnitude is due to a gradual increase in calcium ions in the sarcoplasm, as the ion pumps in the sarcoplasmic reticulum are unable to recapture calcium ions in between stimulations. So what's happening is when we're doing these multiple stimulations, we stimulate, calcium ions have to be recovered in order for a full relaxation to take place. But now think of it, we apply the first stimulus, calcium leaks out, calcium starts to be recovered, but before all the calcium can be recovered, we stimulate again, so even more calcium leaks out. We have even more calcium binding to troponin, uncovering even more active sites. So the myosin can ratchet and pull even more forcefully against the actin and create even stronger contractions. I hope that makes sense. Okay. So moving on then, we can take a look at our next type of contraction, which is called a tetanus contraction, again, based upon how we're stimulating the muscle fiber. Tetanus contractions result when the muscle receives a series of stimuli in very rapid succession. During tetanus contractions, the muscle will remain in a constant state of contraction. There are 2 types of tetanus contractions. The first type are what we call incomplete or unfused tetanus contractions. During incomplete or unfused tetanus contractions, the muscle partly relaxes between the application of the stimuli. The other type of tetanus contraction is called a complete or fused contraction. Complete or fused tetanus contractions occur when the stimuli are applied so quickly that the muscle doesn't really have any chance of a relaxation phase at all. As soon as it hits that relative refractory period, boom, we're stimulating it again, and it sort of stays in a constant state of contraction. So we should note here that all body movements, even the blink of an eye, are the result of tetanus contractions. As a matter of fact, the most common are the incomplete or unfused tetanus contractions. Complete or fused tetanus contractions generate a tremendous amount of force, but the muscles would poop out too quickly to maintain body movements for a very long period of time. So most of the tetanus contractions are of the incomplete or unfused type that result in body movements. Again, back to our graph here, there's our single twitch contraction. So here would be, we could put summation contractions in. So in order to get the summation, we would have to stimulate the muscle fiber around 20 to 30 pulses per second. If we stimulate at 20 to 30 pulses per second, we're going to get a gradual increase in magnitude. But we can see here that we're well into the relative refractory period. On the other hand, with our incomplete or unfused tetanus contractions, we would stimulate at a rate of about 30 to 50 pulses per second. Now, if we're looking at the nervous system then would be stimulating the muscle at a rate of about 30 to 50 stimulations per second. This will result in incomplete tetanus, and with incomplete tetanus, we would get to a point where we'd have enough magnitude or pulling force to actually create body movement, and then it would kind of flatten out at that point. If we wanted to get to fused or complete tetanus, we'd have to go more than 50 pulses per second or more than 50 stimulations per second. But that would increase the magnitude and result in even greater pulling force. But again, it's going to burn ATP supplies very, very rapidly, so it can't be maintained for very long. So it's not an effective method to create body movements. This would be something, let's say, for example, if somebody, went out and we saw them, I remember a scenario a few years back where a girl went out and her father was working underneath the car and the car fell on top of him. She saw it and she ran over, picked up the bumper of the car and lifted almost superhumanly the bumper or lifted the car off of her father. Well, that's going to involve hormones, but also that would require kind of the force that would be generated by complete tetanus contractions. And by the way, there's our single twitch. Single twitch contractions, if we are under 20 pulses per second, we'll just get complete relaxation between, and we really won't get any increase in magnitude. Okay, that takes us to a couple considerations of muscle contractions and things we want to talk about just very quickly as we wrap this up. So the first term is contracture. Contracture is an extremely slow relaxation of a muscle due to overstimulation and the accumulation of waste. So remember, in order for a muscle to relax, the actin has to slide back over the myosin. But if we stimulate the muscle over and over and over again, what can happen is we can accumulate waste products inside the muscle fibers, inside the myofibrils. And those waste products might be lactic acid, free creatine, so on and so forth, and ADP, ATP. But these will then all act to inhibit the ability of actin to slide back out over the myosin. So the actin might tend to slide, even though we've in the relaxation phase, very, very slowly back out over the myosin. The muscle, we would say, is in a state of contraction. Next consideration we have is fatigue. Fatigue is the inability of a muscle to contract or respond to a stimulus, and this is due to the depletion of necessary chemicals like ATP and creatine phosphate. If you just don't have enough energy supplies, the muscle can't contract. The muscle is said to be in a state of fatigue. Finally, and this will be important as we move into the cardiovascular system, we want to consider Starling's law. Starling's law says that a muscle will contract with even greater force when its muscle fibers are stretched. So why does the muscle contract with even greater force when the fibers are stretched? Well, it has to do with troponin and its affinity for calcium. Whenever the muscle fibers are stretched slightly before the contraction takes place, this increases the affinity for the troponin. So with that increased affinity, whenever the calcium leaks out of the sarcoplasmic reticulum, even more calcium is bound to troponin, which exposes even more binding sites for the myosin cross bridges. more myosin cross bridges can bind to the actin, therefore more pulling elements pulling the actin in over the myosin, creating a greater force of contraction. This will be very important whenever we take a look at the cardiovascular system and we talk about heart muscle contraction. So I want to introduce that at this point. Well, we've come to the end of this PowerPoint. We'll stop there for now, and we'll look forward to the next lecture screencast. Have a good day, everyone.